It's our pleasure to introduce our first keynote speaker, who is Mohsen Ghanbari. He is visiting us from Erasmus Medical Center. He is a physician who has worked in the clinic and has training in molecular biology and epidemiology. So this gives him a really great background to perform his multidisciplinary work, where he looks for genetic markers for age-related diseases, which are very complex. So we are excited to hear from him today about his work with microRNAs, I believe, or non-coding regions of RNA and age-related diseases. So if you could please come forward to hear your talk. I also saw that he recently had work coming out in Nature Communications about the relationship between DNA methylation and epigenome regulation and coffee consumption. So this is something we can also ask him about at the break.
Okay, good morning, everyone. Thanks for your kind introduction, first of all. It's a great pleasure for me to be here today and present some of our project in this festival. But first of all, I would like to thank all the organizers for inviting me and giving me the opportunity to talk about this, let's say, emerging topic. So in recent years, the availability of large-scale biobanks and omics data has grown rapidly, and in many countries, actually, you see the biobanks initiatives or large-scale large-scale omics data that are available now, publicly available, but it's still a bit challenging to integrate different omics layers from population-level data that maybe they can help us for the better understanding of the etiology of disease and identify novel biomarkers for complex diseases. So that's the reason I'm going to talk about this, let's say, complex topic, population-level omics data integration in the context of non-coding RNAs, which is also an emerging field in biology because of their crucial role in many biological processes and in the etiology of many diseases. So with the diverse audience that we have in this festival, I would like to start with some very basic concepts, and then we will dive a bit into the topic with some examples of our work. Okay, so first of all, we know that the human body consists of different cell types, such as neurons, cardiomyocytes, or osteocytes, that all these cells, they have actually the same genetic structure, instructions, but they have different structures and functions. So if you have the same genetic blueprint for all these cells, why we see different cell types? One reason could be epigenetics, because we have epigenetic regulations in the embryogenesis stage and also later in our life that can actually change the pattern of gene expression, and you see a tissue specificity for gene expression. About epigenetic, so it's the external modification to DNA that turns genes like on or off, and these modifications actually change the DNA sequence. They don't change, the DNA sequence, but they instead change the gene expression through actually affecting how cells read the genes. So we have three main epigenetic markers: DNA methylation, histone modification, and RNA interference. These are, let's say, the most well-known epigenetic markers in the field. Starting from DNA methylation. So DNA methylation is the, let's say, the most characterized and the best studied epigenetic marker in the field. So it's a biological process by which a methyl group is added to a cytosine base and change the activity of a DNA segment. So we know that DNA methylation can change the pattern of gene expression by changing the activity of promoters, enhancers, and also other regulatory elements. But during life also, there are many lifestyle factors and environmental factors that can change the DNA methylation pattern. So we, and of course many other researchers, they have shown the impact of smoking and alcohol consumption on the level of DNA methylation and changing gene expression which I'm not going to talk about this in much detail. As a colleague mentioned also, we recently also published a paper that's a complex paper. We did it in 16 different cohorts. At the population level, we showed that how coffee consumption also can change the pattern of DNA methylation and also can change your risk for many metabolic diseases, including type 2 diabetes, fatty liver disease, or let's say obesity in general. But the good news is that that's actually a kind of a positive effect. The amount of coffee that you drink in a day can increase your metabolism rate and then can reduce the risk of many metabolic diseases. But that's about the first epigenetic mark. But actually, that's the focus of my talk today. I'm going to talk more about the RNA interference or gene silencing by RNAs. So we know that gene expression is also regulated by different types of RNA. This is a figure I think that all of you know from the central dogma. We have genetic information, that these genetic information can be transcribed to RNAs, and then these information on RNA can be translated to build proteins. But actually, this figure is not completely true because there are many RNAs that are not translated to proteins. And they, of course, that they are involved in protein synthesis and gene regulation, but they are not translated to proteins. So if you look at the human genome, we also see the whole genome, whole transcriptome data, for example, from ENCODE project, we see that more than 70% of the human genome is transcribed into RNA molecules. But less than 5% of the RNA have the potential to encode for proteins, which is actually, some people say that something between 2 to 3% of RNAs can be translated to proteins. And actually, there are also some other studies that they show the kind of link between the complexity of an organism and the amount of non-coding DNA sequence that we have. So, for example, if you compare the E. coli, a bacterium, to human genome, you see that the amount of non-coding RNA in humans are something like 95 to 98%, but in E. coli, this is much, much less. So with this, I want to introduce then the different types of RNAs that we have. At the most general level, we can categorize RNAs to protein-coding RNAs and non-protein coding RNAs. Protein-coding RNAs are also known as messenger RNAs, and then for non-protein coding RNA, we have housekeeping RNA, non-coding RNAs that include transfer RNA and ribosomal RNA, and then regulatory non-coding RNAs that based on the transcript size, we can further categorize them to long and small non-coding RNAs. And among the small non-coding RNAs, we have microRNAs that are, again, let's say, the most characterized non-coding RNAs so far. So about microRNAs, they are small single-stranded form of RNAs, highly conserved in different species. And we know more than 2,000 high-confidence microRNAs just in human, that they can regulate gene expression at the post-transcriptional level. And based on the in silico analysis, we know that more than 2/3 of all coding genes are like a potentially putative targets for microRNAs. So microRNAs, they were discovered almost 3 decades ago, but during these 3 decades, their importance in gene regulation has steadily gained a kind of appreciation, which is also indicated in the number of publications that you see in, especially in the last decade. And now we have evidence for the involvement of microRNAs in almost every physiological and pathological processes. I can summarize the importance of microRNAs in, let's say, 3 main characteristics. They are molecules that they can provide deeper insights into the disease etiology. They are detectable in many biological fluids, like blood, let's say urine, saliva, and they can be used as potential biomarkers for diseases. And they have also the potential to be used as therapeutic targets. About the biogenesis and function of microRNAs, so just in brief, after transcription in the nucleus, so they can be processed by two enzymes to form two pre-microRNA structure, and after transferring to the cytoplasm by exportin 5, then we have a mature microRNA, which is actually after processing by dicer enzyme, and then at the end we have a single strand small RNA that can bind to the 3' UTR of messenger RNAs and degrade messenger RNA or repress translation. So in brief, microRNAs are involved in gene silencing. So I mentioned that there are many actually papers that they show the importance of microRNAs in different biological processes and in pathogenesis of many diseases. What you see in the field, usually people start with some number of, let's say, healthy or disease samples that can be cells and tissue, and then after RNA extraction or doing RNA sequencing, then you have a list of RNAs that are upregulated, downregulated in disease condition compared to the healthy controls, and then you end up with a list of microRNAs that are also upregulated, downregulated, and after the correlation between the expression of these two microRNAs and messenger RNAs, you can make a matrix, and then, of course, that you do validation studies to confirm your findings. But this is what we have more in biology. So a couple of years ago, we actually introduced a model that you can maybe use population-level data. I say that there are many biobanks or many, let's say, multi-omics data available at large scale that you can use them actually to detect maybe microRNAs for complex diseases. So with the availability of genomics data, transcriptomics data, proteomics, metabolomics data, and if you can combine these data sets, maybe you can find valid biomarkers or valid molecules that are involved in the etiology of complex diseases. So I introduced just one example that when we started with almost 10 years ago with this model. So we saw that if in the normal situation, if a microRNA binds to the 3' UTR of a messenger RNA, then the translation is repressed, and then you have lower amount of protein. But if in the disease condition, if you have a genetic variant located in the microRNA encoding sequence or in the binding site, then the microRNA cannot actually bind to the messenger RNA and cannot do its function. Then you have, let's say, a bit more protein. And having too much protein, if the protein is harmful, then can alter the risk of diseases. So that's one example. Actually, that's the number of, let's say, SNPs that we have. The total number of common SNP, these are common SNP, of course, that we have many more rare variants that you cannot detect them, let's say, with the genomics data that we have, let's say, using GWAS data, because we are focused on the common variants. So we have, let's say, 250 variants that are located in all sequences that encode microRNAs, if you have more than, let's say, 2,000 high confidence microRNAs. And that's the number that you see, the genetic variants that are located in the 3' UTR of target genes of all microRNAs. So the variants can be located somewhere here in the microRNA sequence or in the binding site. So that's the approach. So we start with the genetic data in microRNAs or binding sites, and then combine it with the very large scale GWAS data to find some candidate, and of course to prove the functionality of those variants by doing some experimental validation studies. So this is the example that about a microRNA that we discovered actually almost 8-9 years ago. This is the first report for microRNA 4513, the association of this microRNA with cardiometabolic traits. So we combined the SNPs in microRNA binding sites or microRNA encoding sequences with several GWASs, large scale GWASs, something like with the sample size more than 100,000 samples in the field of cardiometabolic traits or diseases. And we found a SNP located in the nucleotide number 8, which is a crucial part of a microRNA for binding site recognition. And we found the association with diastolic and systolic blood pressure, the serum level of cholesterol, LDL cholesterol, fasting glucose, and coronary artery disease. In particular, it was very significantly associated with blood pressure. So then we decided to do some experiments to actually test if that SNP can really change the expression of microRNA or can change the function of microRNA, because the SNP is located in the seed sequence of microRNA, which is crucial for binding site recognition. So by doing like a vector analysis, we cloned the microRNA sequence with and without mutation in cell culture, and after like almost 2 days, we measured the level of microRNA, and we found that indeed when you have a mutation, the expression level of the microRNA is reduced. And then in the second situation also, we test if the microRNA binding to its target gene can be changed when you have the variation. So we did the experiment using luciferase reporter assay, and then we showed that at least for the top target gene, which is GOS-R2 for blood pressure, that's a very known gene for atherosclerosis, we found that the interaction between microRNA and the 3' UTR of that target gene is changed when you have the G allele or A allele, and G allele is actually the major allele, A allele is the alternative allele here. So with this, we showed that, okay, if you start with the genomics data, we could show that the SNP can change the expression of this microRNA and also can change the binding of this microRNA to GOS-R2 gene, which is important for atherosclerosis of blood pressure. And actually, that was a kind of explaining the reason for the association between this SNP, which was identified by many GWAS for blood pressure. But if you back to the GWAS results, you always see that there is no good annotation for this SNP, and people just report this SNP as a non-coding SNP. So after this project, actually several other papers published, maybe I think more than 50 papers published about the importance of this microRNA. But this is also a very interesting project that other groups, they show that the SNP actually cause a kind of allele-specific target gene regulation of this microRNA by doing gene expression analysis. They did RNA sequencing and PCR for all genes, and they showed the number of genes that are differentially, let's say, regulated when you have two different situations, a major allele or minor allele. And also they confirm it with protein analysis, with Western blot, and also luciferase reporter assay. So by that story, we actually started this project that I want to actually present today, which is a comprehensive project that's a cross-omics data integration to identify the genetic regulation of microRNAs and disease association. I'm going to explain a bit about each of these steps in my next slides. So in the first step, we measured the level of plasma circulating microRNAs in the Rotterdam study with in almost 3,000 participants using a targeted RNA sequencing approach. And I say why we measured the level of cell-free or circulating microRNAs, because in many studies, you see that the microRNAs can be actually transported to extracellular matrix by different mechanisms in disease conditions. That means if you have a problem, if you have a disease in the brain, in the heart, or in other tissues, you have microRNAs that are upregulated or downregulated in body fluids, and then that can be used as biomarkers. And you can also see in many studies that these molecules are relatively stable to be used as biomarkers. So there are many approaches that you can detect microRNA levels, like qPCR or arrays or other types of techniques, but we used a specific, or let's say targeted RNA sequencing approach introduced by HTG company, which has been shown to have a kind of high reproducibility and specificity for microRNAs. So with this approach, we measured the level of 2,083 microRNAs, and then we did a GWAS on the level of each single microRNAs. That means in total, we linked the level of each microRNA in plasma to more than 10 million SNPs that we have in our genome. Let's say linking genomics data to transcriptomics data or microRNA data here to identify SNPs that they may change the level of microRNAs in a hypothesis-free approach. Yeah? And then we replicated our findings in 2 cohorts, Ottawa study and Framingham Heart study, that you can see the number of participants, in total something like 8,000 participants, and the number of microRNAs that we have in these 2 sets. Actually, we have like more than 2,000 microRNAs. So that's the results of the first part. So you see, based on the position of SNPs in relation to the microRNA sequence, if it's in the window of 500 kb upstream or downstream, that's the kind of accepted, let's say, the region for eQTL analysis. You call them cis elements or cis eQTL. And if the SNP is located far away from this, the sequence of microRNA, you call them trans microRNA eQTLs. So in total, in this study, we found more than 4,000 cis and trans microRNA eQTLs for 64 microRNAs at a very conservative p-value, which is based on the number of SNPs that we tested, almost 1 million independent SNP times to the number of microRNAs that we have here, more than 2,000. Okay, then in step 2, we actually link the association, or we test the association of microRNA eQTLs with diverse clinical outcomes that we have, the GWAS catalog. In the GWAS catalog, you see a list of all SNPs so far reported for any traits or clinical outcome. So we additionally did the co-localization analysis that when you have the data from eQTL and you have the data from a GWAS, so you try to see if the top variant that you have identified based on the eQTL here, microRNA eQTLs, so it's a SNP that is reported for any clinical trait or outcome at the end. So this analysis actually showed cis and trans microRNA eQTLs to be associated with like a diverse clinical outcomes like blood pressure, cancer, mental health, hematological traits, anthropometric, and other traits that I have mentioned here for trans eQTL. And also you can see in this circular plot the microRNAs that are linked to different clinical outcomes. Okay, but this is only kind of association analysis. We still need to prove it using validation studies, or we have also an approach in our field that we do Mendelian randomization that I will show in the next slide. But before going to the Mendelian randomization part, I want to also show the analysis that we did using UK Biobank. So in UK Biobank, I think it's like familiar to all of you that in UK Biobank we have more than 425,000 participants with genotyping data. And then the clinical outcomes that are reported in UK Biobank, there are 900 clinical outcomes that we filtered them based on having more than 200 cases in each that can be categorized in 16 disease categories. And I should say why we did this analysis, because I said that microRNAs can bind to many target genes. We have microRNAs that they have more than, let's say, 200, sometimes 500 target genes. Then you can imagine that microRNAs can be involved in different diseases, different biological processes. That's the reason that we usually check also for pleiotropic microRNAs. So with this data set, which is publicly available, we did a phenome-wide association study. That means instead of taking genes or the whole genome, you take the variants. So you have 900 phenotypes, and you have SNPs that are associated with the level of, let's say, a microRNA. So with 900 phenotypes, you can run the analysis for that single SNP, which we call it PHIWAS, and then check if there are microRNAs that are associated with multiple traits. So based on the single cis instrument analysis, which is just taking one SNP, for 85 microRNAs, we found 9 microRNAs that are pleiotropic, and they show association with many clinical outcomes. and calculating genetic risk score, again, based on the CIS instrument for 119 microRNAs, we found 16 microRNAs that they show association with multiple traits, and you can see some of these microRNAs here that they show association with many traits. But as I mentioned earlier, so these are just the association. We need to prove them. So one approach that we usually use in our field, in genetic epidemiology, is checking for causality, which is a Mendelian randomization approach. You have instrumental variables, you have exposure and outcome, and here actually instrumental variable are your genetic instruments. The SNPs that they show association with an exposure, here is microRNA, and then you have a clinical outcome. And actually these SNPs should work. There are some assumptions for Mendelian randomization study that the SNPs should work actually through the exposure and not through the other ways to change the risk of disease. So this analysis actually showed us, at least for some microRNAs, we see a causal association. That means these microRNA might be causally involved in the etiology of diseases like MIR1908, which is causally associated with colon cancer and other microRNAs that you have here. But these microRNAs are, let's say, candidates for further validation or experimental studies in vitro or in vivo to prove their role in the pathogenesis of these diseases. Okay, next, that's the additional analysis that we did to link microRNAs to different omics data. So we know that there are many studies that they check for the association between genetic variants with the level of the different omics layers that we have. For example, the association between SNPs and the protein with the gene expression level that we call it EQTL, expression quantitative trait loci, or the association between SNPs with the protein level, we call it pQTL, or with the metabolite level, mQTLs. So here we take the genomics or the genetic data as a core and try to link other omics data to microRNAs to maybe find pathways that are regulated by microRNAs. So we focus on the SNPs that are associated with microRNAs, but which are also reported in other studies, large-scale studies, to be associated with any of these molecules and also with risk of diseases. So as you can see here, so we have 64 microRNAs, 4,000 EQTLs for 64 microRNAs, and the sample size that we have is 8,000. So we get the data from a larger scale gene expression, quantitative or EQTL data with 32,000 sample size, and we found 39 microRNAs that they show association with 164 genes. If we do it with the protein EQTL data with a sample size of 30,000, you see 29 microRNAs are associated with 112 proteins. And also with the metabolites, 10,000 metabolites, 13 microRNAs with more than 200 metabolites that are mainly from Nightingale and Metabolon platforms. So again, these are all based on the blood level of these omics. Of course, that if you have access to other tissues or the tissues related to the phenotype of interest, then you need to check it. But it's a bit difficult because if you work with the large scale sample size, that's what we have in the population studies usually. That's the blood samples. So the results of all these steps are actually summarized now in an atlas that we generated for the genetic regulation and disease association of microRNAs that we call it ADMIRE Atlas or miRNAs. That's a term that we like, and we have added actually to this setting to link genomics, proteomics, metabolomics, epigenetics to miRNAs. Let's say that's a new omic. And actually the results are now publicly available. So we have submitted this paper in medRxiv, but you can go to this website and search for genetic regulation of microRNAs, disease association, or omics integration and the data are all publicly available. So I have one example, take one example from this web tool to show how a genetic variant in a microRNA can change the risk for Alzheimer's disease. So for people who are not familiar with Alzheimer, so the late onset Alzheimer disease is manifested by progressive loss of memory and cognitive decline. So amyloid plaques and tau tangles, they are in the brain, they are the two hallmarks or the two main mechanisms of Alzheimer's disease. But of course, that the larger scale GWAS, they have also introduced or they have identified more than 50 risk loci for Alzheimer's disease that together can explain almost 30% of the disease. So this is one example of a GWAS on Alzheimer's disease. This will be the large scale GWAS with the sample size more than 450,000 participants. And yeah, you see that the different steps here, but many of the analysis, many of the SNPs that the people they have reported in this larger scale GWAS, they are non-coding SNPs that I said earlier in the talk that for non-coding SNPs or non-coding variants, usually we don't have any clue for their function. But when we run a GWAS or when we report the genome-wide association study for a trait, we try to annotate the SNPs to the nearby coding genes. But we usually ignore the other non-coding elements that might be important for the disease. So that's one SNP located in chromosome 17 in two different GWASs. Larger scale GWASs, you see that the SNP is top SNP, and people, they have reported BizRAP1 as the annotated gene. There's a kind of zoom in on the region. This region. So here you see 4 SNPs that are in high LD, and they are actually inherited together. And these are the 3 genes or like a gene non-coding RNAs or non-coding genes that are located in this region: BizRAP1, antisense, and miR142. And the EQTL data show the association between this SNP and the level of miR142 in blood. So we checked the position of this SNP, and we found that actually the SNP is located exactly in the promoter region of miR142, and this is the data that we extracted from Phantom project and other projects that actually they show the position of promoter region of all miRNAs in our genome. And we did the experimental studies, like in vitro experiment. We cloned the promoter region of the GFP with and without SNP, and then checked the promoter activity. And you see that there is a kind of difference, which is still significant, for the activity of the promoter of miR142 when we have the reference allele or alternative allele in the position. So we did 2 experiments to prove our finding and to show also if the miRNA is really important for Alzheimer's disease. So first, we overexpress the miRNA in iPS-derived neuronal progenitor cells, which is, let's say, a relevant cell for Alzheimer's disease, in 3 different conditions: untreated cells, overexpression with a negative control, and then with miR142 miRNA mimic. And after doing RNA sequencing, we found several genes that are downregulated in the situation that we have overexpression of miR142. You see the number of target genes. These are 2 strands, 3P and 5P. There are 2 strands of miR142, and we did 2 experiments for each of these strands, and see the number of target genes. Next, we did the additional analysis in knockout mice in 3 different conditions, and we did the RNA sequencing from the hippocampus of these mice, which hippocampus, again, is the target tissue for Alzheimer's disease, right? So, and then we extracted the list of genes that are upregulated when we have like a knockout situation. And if we compare these 2 lists, upregulation of miRNA artificially and in mice model, you end up with genes that are really important for the risk of Alzheimer. I have mentioned two genes here, TGF-beta receptor 1 and PCOM, that are both among the important genes identified by GWAS for Alzheimer's disease. So as I said, we started with SNP located in chromosome 17. We linked these data with eQTLs, and we found that the SNP actually can change the promoter activity of microRNA and reduces the expression level of these microRNA. And when you have lower amount of these microRNA in the brain, there are genes that are upregulated, and these genes are important for brain function. And then if you have more expression of these genes, then the risk of Alzheimer is reduced. So this is what we have published actually in this paper, but additionally we did another project, and we did it in details what's happening in the brain exactly when we have miR142 3P, and we showed that the microRNA is involved in the tau pathology. I'm not going to talk about this project, but in general, that's our finding for this microRNA. But interestingly, two other projects or two other papers, they also show the importance of this microRNA as a biomarker for Alzheimer's disease. So in the first paper, you see that the authors, they did microRNA profiling from the hippocampus of 80 patients. Hippocampus is the target tissue, right? So, and the microRNA 142 is among the 15 microRNAs upregulated in the hippocampus of 80 patients. And in the second paper, the authors, they have checked the level of this microRNA. Actually, they did microRNA profiling for all microRNAs in CSF and plasma. And again, miR142 is among the upregulated microRNA in 80 patients. So that's a kind of proof that this microRNA can be used also as a biomarker for disease. So these are, in general, about the story of cross-omics integration at the population level for non-coding RNAs. And so I think I have one more slide for challenges, because you mentioned about the challenges that we have in multi-omics data integration, in particular for population level data. So you are, I think, familiar with many of these challenges. Maybe for the sample size, now we have, let's say, less problem with the availability of many biobanks and like different omics data sets that are available through these biobanks. But still, the target tissue is important, because what we have from population level data is mainly blood. And if we want to link it to the clinical outcome, we need the target tissue. There are some ongoing projects that how we can, for example, generate iPS cells from blood samples of participants in cohort studies to go and to like differentiate those cells to the real or to the right target cells. But there are kind of ethical and privacy issues that we are faced now for this kind of project. But data quality is important. Because you can imagine that the omics data can be generated through different approaches, and it's not always like a harmonized through different cohort studies. When you work with a large scale data, you want to combine data and do meta-analysis with several cohorts, but that's also an issue that we have sometimes. Maybe not for genomics data, which is cheap and everybody can do it with the genotyping. It's the kind of standard approach, but for other omics, for like metabolomics, for proteomics, for gene expression, so it's still kind of issue that we have. Of course, that for the integration of data, it's a complex approach that you want to combine different layers with the different nature that they have, or the number of molecules that they have. These are some still issues that we have in the field. And of course, that for the interpretation of the data, when you have results from different layers, when you want to combine these results and make a good story for your hypothesis or for the disease, it's still difficult. And of course, that I mentioned earlier, that for the cohort studies, we have also some ethical or privacy issues that, for example, for the consent informed, we have, let's say, in many participants that they are entered to, for example, our cohort in Rotterdam, They started almost 30 years ago. They have died, or we have not collected the right constant info to, for example, generate iPS cells from the blood cells. So these are some like issues that we have in the field, and also for sharing data with other sources, but we are trying to manage them. Yeah, thank you very much, and I'm happy to take your questions.